Up to this point in the class, we have talked about individual cells and molecules within individual cells and chemical reactions within individual cells. In Chapter 9, we're going to look at communication between cells, whether those are single cells in a solution or cells that are in contact with each other in a multicellular organism. We will do this in 2 chapters. This lecture will be the first part and will deal only with eukaryotic cells. Different types of signaling that can occur in terms of very broad categories are autocrine, so self-signaling. This cell has secreted this molecule, and that molecule signals back to the cell that secreted it. In cell signaling across gap junctions, and if these are gap junctions, those are in animal cells, this signaling cell is connected by gap junctions to the target cell. And so the green cell makes the signal, and that enters the red cell and causes some sort of effect. And that would be nearby cells, and that's across the gap junction. We can have paracrine signals where these 2 cells are nearby each other, but they are not connected. They're not part of the same tissue. And so this signaling cell is releasing chemicals that are received by a very close by cell that is called paracrine, around in the same neighborhood. Endocrine signaling, the cell that is secreting the signal is somewhere quite distant to the target cell. And one of the first examples that comes to my mind would be like our human growth hormone is secreted by our pituitary gland that's in our brain, but those chemical signals go to cells all over the body to have their effect. That would be an endocrine signal. Hormone signals are endocrine signals. They are created in cells, but their effects can be felt by cells quite distant from the originating cell. This is showing a nerve cell, or 2 nerve cells. There is the presynaptic and the postsynaptic cell. So this space between them is the synapse, right? Before the synapse and after the synapse. This cell is the one that is secreting the signal. In this case, a neurotransmitter, because neuro means a nervous tissue. It's a transmitter because it is sending the signal away from the originating cell. That is released into this space between the cells and is received by the postsynaptic cell. That cell has receptors on its surface. Those are going to be proteins attached to the cell membrane of this receiving cell. And the neurotransmitter is the signal. The shape of that signal molecule fits perfectly into the outermost shape of the receptor. And the signal is called a ligand, and the receptor is what the ligand binds to. So the receptor molecule is almost always a protein, and it is that which binds the signal. The signal is called the ligand, that which is sticking to the receptor. This example is showing neurotransmitters, but this happens in other types of cells. And because these 2 cells are very close to each other, but they are not connected by gap junctions, this would be an example of paracrine signaling. We don't have to have a signal on the cell surface. This is showing the membrane of the nucleus. This is the plasma membrane, so the cell membrane. This is the fluid outside of that cell. This receptor molecule is called an intracellular receptor because it is actually inside the cell. It is not sticking out on the surface. The signaling molecule that has been secreted possibly by another cell in the case of a paracrine signal could be an autocrine cell signal if it came from this. In either case, the receptor in this situation is actually in the cytoplasm of the receiving cell. And once again, the shape of the receptor fits the shape of the ligand, the signal, and that can be brought, in this case, into the nucleus and can interact with the DNA that can control gene expression. I've labeled the signaling molecule as hydrophobic because if it is hydrophilic, it would be repelled by those hydrophilic heads of the phospholipid membrane. If it were very large, it would need some sort of transporter. So the only way it's going to diffuse across the membrane is if the signal is small and hydrophobic. When this combination of the signal binding to its receptor and then cross the nuclear membrane, this signal is going to interact with the DNA, and that is called a transcription factor, because when it binds to the DNA, it is going to affect whether or not the cell can use enzymes to read that DNA and transcribe it to make the RNA. This is showing a gated ion channel. It is a transport protein. It is gated because it is closed off and will not allow passage unless the signal molecule is received. The signaling molecule is not what's getting transported. The signaling molecule is binding to that channel. When that signaling molecule binds, that triggers the gated channel to open, and then the ions can cross. If we remember back to our chapter 5 about the structure of membranes and what can and cannot flow freely across them, ions are charged, and charged particles cannot diffuse freely across a membrane because of the membrane potential. A gated ion channel is one way that those ions can cross, and there is a cellular signal that triggers this gate to open and allow the diffusion, the facilitated diffusion of these ions across the membrane. This is one of the more complicated eukaryotic cell signaling processes. It is called G protein coupled receptor, and that's gpcr, G protein coupled receptor, yes. I will walk us through this diagram, but I have also posted in the module along with this lecture several links for specific examples of how cells use these G protein coupled receptors. They are very popular and very often used mechanisms of signaling in cells. They are also targets for drug therapy. They are called hetero, trimeric. Meric, as in monomer or mer, is unit. Tri is 3, so there are 3 units. And if it's hetero, means there is a mixture. The 3 units are not the same. The 3 units are alpha, beta, and gamma. It looks like a Y, but that is a lowercase Greek gamma. Here is what it looks like in the cell membrane. So this is the phospholipid bilayer. This whole thing is the G protein coupled receptor. So the green blob is the receptor. The G protein is coupled to the receptor, bound to the receptor. And there is the alpha subunit, the beta subunit, and the gamma subunit. We also see this blue blob labeled GDP. That stands for guanosine diphosphate. It is energetically related to adenosine diphosphate. In our metabolism, we look at phosphate being added to ADP to make ATP. Here, we're going to see a phosphate added to GDP to make GTP. Guanosine diphosphate, guanosine triphosphate. Same energetically in terms of the 3 phosphates that are attached, but instead of an adenine, we have a guanine base. So the GDP is associated with the alpha subunit. Only the alpha unit is bound to the GDP. When a signaling molecule binds to the receptor is not the G protein, the receptor is coupled to or bound to the G protein. The signal molecule binds to the receptor. When that happens, that triggers a GTP to replace GDP. We're not exchanging phosphates here. The whole GDP comes off and the whole GTP comes on. When GTP is bound to the alpha subunit, that triggers the beta and the gamma subunits to come off. They are still adhered to the membrane by the gamma unit, but they are separated from the alpha. The alpha subunit is still attached to the receptor, and this is what triggers some response inside the cell. And there are many examples that are put in the module for those types of responses. There are a lot of different responses that could happen. Once the response has been triggered, then we have a phosphate removed enzymatically from the guanosine triphosphate. If we take a phosphate off of guanosine triphosphate, we restore back to guanosine diphosphate, and that is the signal for the alpha, the beta and gamma subunits to reattach to the alpha, and we are ready to repeat this signaling pathway. It's a lot. That's why I've provided several examples in the module to go along with this very static picture. Another common type of receptor in eukaryotic cells is called an RTK signal. That stands for receptor tyrosine kinase. It's Topo Chico. It's fizzy water. Receptor means it's going to bind to a signal, some ligand that will stick to it. This crosses all the way across the plasma membrane, and it is dimeric, 2 pieces. It has only one space where it crosses the membrane, but there are 2 separate ones. That when the signal binds, then these 2 individual units associate with each other closely in the membrane. And the tyrosine residues, that means the tyrosine amino acids that are part of that receptor and on the inside of the cell, they're intracellular, are autophosphorylated. They trigger the response that causes phosphates to be attached to them. Phosphorylation of the tyrosines on the inside of the membrane will trigger a response in the cell, and that signal stops when a phosphatase removes the phosphates, and they go back to looking like their original location. The signal comes off, and the individual components of the receptor tyrosine kinase dissociate and are still next to each other but not interacting. We saw steroid hormones when we were looking at our macromolecules, and in particular our lipids. So steroid hormones are lipids. And this is a reminder that lipids are not polymers because there's not a single type subunit that links to itself over and over again to build the large molecule. Lipids are grouped together as macromolecules because they are big, and they are primarily hydrophobic. They are water-hating. They have small areas that are polar, amphipathic, they have both, but they are tiny, tiny functional groups compared to the rest of the molecule. So there is estradiol, that is a steroid hormone. That's testosterone. They have the same number of rings, but their functional groups are different. Here we do have the hydroxyls and the methyls attached to 5 and 6 carbon rings, but estradiol has another hydroxyl, whereas testosterone has a carbonyl carbon. It would be a ketone because it's in the middle of the molecule. Estradiol also has more double bonds in this 6 carbon ring, and they are in a different position than the 1 double bond. I will not put these up and have you identify which is estradiol and which is testosterone, but shown a picture, you should be able to identify that these are steroid hormones. I'm going to move myself so that we can see what I've written on the last one, and that is cholesterol. It has a very large hydrocarbon chain that sticks off of its 5 carbon ring. It has double bond in this ring as opposed to that one, and it has the single hydroxyl functional group. That is the only region that is polar, and cholesterol is that lipid, that's large lipid found in the cell membrane of animal cells, and it's just this little tiny hydroxyl that allows this molecule to interact with the polar heads, either facing toward the inside or the outside of the cell, and the rest of the very hydrophobic cholesterol molecule is embedded in those hydrophobic fatty acid tails. Steroid hormones are going to be endocrine signals created in some cell and having their effect at cells that are quite distant from that. They are hydrophobic, so they could diffuse across that plasma membrane to interact with internal receptors. They don't require a cell surface receptor. They can diffuse across cell membranes. I don't know if there's any really good place to put myself right now. Scooch me down here for now. This is showing epidermal growth factor. The next few slides are going to have a lot of abbreviations, and I've done my best to make sure that I've defined those abbreviations. If I miss any, I will try to get that corrected. Epidermal growth factor is EGF. Epidermal is our skin, our epidermis. So growth factor, it's what is a signal that if we damage our skin, we can make new skin cells. Sorry if this is an awkward transition. I had a sneezing fit. So our epidermal growth factor, helping us to repair damaged skin, was also very important when we were developing as fetuses to create all of the skin cells that we need. there is an epidermal growth factor receptor. Epidermal growth factor is the signal or the ligand. EGF is the ligand, so EGFR is the receptor for epidermal growth factor. This is a specific example of a receptor tyrosine kinase. So if the epidermal growth factor binds to the epidermal growth factor receptor, that causes it to come close together and dimerize, create the two hanging out. The tyrosines on the inside of the part of the receptor facing the inside of the cell become phosphorylated, and that triggers a series of reactions that tells the cell to grow and divide. So for our cell growth, wound healing, tissue repair, you get scratched, cut when you have some sort of wound that needs to be sealed back up. That is going to trigger your epidermal growth factor to bind and tell that cell and its neighbors to make more until we have repair, and then that signal will stop. If the signal does not release, then we get continual information to the cell to keep dividing, and that can cause tumors. So if our epidermal growth factor binds to the receptor, we get proliferation, cell division, mitosis. We get cell migration and adhesion. We're fixing a wound. And it also plays a role in angiogenesis. Genesis is birth, and angio refers to our blood vessels. So that's making new blood vessels. So that is showing that if we have the epidermal growth factor bound, we trigger a series of events that stimulates translation, the generation of proteins, and that is our cell proliferation, migration, adhesion, and it inhibits or prevents apoptosis. Apoptosis is programmed cell death. So if we're needing to make new cells, then we want to prevent cells from being eliminated, killed off by apoptosis. Let's see, slide is not advancing. Phosphorylation, again. Move me out of the way. There we go. This is a phosphate functional group. The phosphorus with a carbonyl carbon and 3 hydroxyls. In solution, it is a hydrogen ion donor, a proton donor. That makes it an acid. And so we end up with these negatively charged oxygens. Phosphorylation, that addition of a phosphate, most commonly happens to these 3 amino acids. There's tyrosine. We saw that in our RTK. Serine and threonine. So the underlined words are the actual amino acid. They all have phospho ahead of them because they get phosphorylated. Phosphorylation, as we've already seen, is really important in cell signaling. Sometimes it's the addition of a phosphate that causes a signal. Other times it's the removal of a phosphate that causes a signal. But phosphates are critically important in cell signaling. This is showing ATP, that cellular potential energy molecule, being converted to CAMP, or CAMP, which stands for cyclic AMP. It's adenosine monoamine. phosphate. This is the adenosine. We have the ribose sugar with an adenine on it. And it is monophosphate because we have only one phosphate. It is cyclic because this phosphate is bound to the ribose in two places, making a nice little molecular circle. This chemical reaction happens due to the enzyme adenylyl cyclase that removes two phosphates, leaving one phosphate attached, and binds this top oxygen also to the ribose. So this is cyclic AMP. The two phosphates that are removed and stay together are called pyrophosphate. This cyclic AMP is also called a second messenger. That means some other ligand or a ligand bound to a receptor. And then this happened, and the cyclic AMP will be the next messenger that will help to activate or inactivate proteins depending on the reaction. Another enzyme dealing with phosphates, phospholipase C. It is an enzyme and it works on lipids, my arrow won't show up, lipids that have phosphates on them. So PIP stands for phosphatidyl inositol phosphate. Again, you're not going to have to recognize that this is a PIP. I would label it. But this phospholipid, here's the lipid, there are the phosphos, is acted upon by phospholipase C, and that generates diacylglyceride and inositol phosphate. And it's inositol 3-phosphate because there are 3 phosphate groups on it. This is important in this chapter because diacylglyceride and inositol phosphate 3 are also second messengers. That means a ligand bound to a receptor that indicated to the cell an activated phospholipase C that generated these two molecules that will then have effects in the cell. This very complicated looking image is showing one way that cells can regulate translation, control whether or not a messenger RNA will actually be read by the ribosome to create proteins. So if we don't worry about all the little blobs piling up over here, in the top image, the messenger RNA has folded on itself and is connected by hydrogen bonds. And that means if a ribosome were to try to bind here, it's going to run into this structure and it can't get past. So we would not get translation. However, we can see that if extracellular activated regulating kinase is present, it is going to signal phosphorylation of two proteins here that are called translation activators. And that will cause the hydrogen bonds to break, the messenger RNA to linearize, and then the ribosome can read along it and create proteins. A kinase is an enzyme that adds phosphates. A MAP kinase is a mitosis activating protein kinase. So a MAP kinase is going to also trigger cell division. So if this mitosis activating kinase, that is the specific extracellular regulating kinase, when it has a phosphate on it, it is going to activate, cause to happen, the translation. Because this chemical will allow for a phosphate to be added to the MAP kinase interacting kinase, meaning it interacts with this MAP kinase, and then when it has a phosphate, it catalyzes the addition of a phosphate to this protein. EIF is eukaryotic translation initiation factor. That is going to allow for protein synthesis. The elongation, that just means the joining of amino acids together to build the protein. That's a lot. The basis is this enzyme, when it has a phosphate on it, triggers the addition of a phosphate to this protein. When it has a phosphate, it triggers the addition of a phosphate to that protein. That causes a conformational change and pressure, physical, mechanical pressure on the messenger RNA to flatten out so that translation can proceed. This is an image of a mouse embryo. Our hands and feet look remarkably like this early on in our development as well, because we start as a solid ball of cells, and then we become a hollow ball of cells, and then we build up tissues and structures from that. Mice are vertebrates, so their embryonic development looks remarkably like ours. How long each stage takes varies dramatically, because the mouse's full gestational age is 27 days. So from fertilization to birth, 27 days. We're still a solid ball of cells at that point. So 27 days is less than a month. But it may take us longer to get to this stage, but our hands and feet start out looking just like this. We have tissue between our fingers or between our toes so that they are webbed. At a certain stage, we get a signal that binds to a receptor that triggers apoptosis. We don't need the webbing between our fingers. We need that to go away, and those cells are eliminated, and that frees our digits, whether it's on our hands or on our feet. If it's incomplete, people are certainly born with more webbing between their fingers or toes. In many cases, it doesn't affect function. It's just incomplete apoptosis. If you look at your hands, you still have some webbing between your thumb and your forefinger. You still have a little bit of webbing between each of your fingers. That's where we apoptose to that point and didn't go any further. And our last example of eukaryotic signaling is yeast. They are single-celled fungi. Because they're a fungus, they are eukaryotic. If they're eukaryotic, the way they divide is by mitosis. These happen to be Saccharomyces cerevisiae, the genus and species that are used in fermenting alcoholic beverages. They typically divide by mitosis and do so by budding. So initially the new cell is very small, and then it will grow to be the same size as its initial cell. But under certain conditions, yeast cells can secrete a molecule called a mating factor, meaning that will trigger other yeast cells not to do sexual reproduction, but to undergo their cell division, that there is enough nutrients in the media for them to divide and to be undergoing additional mitosis. The rest of the chapter is going to be dealing with prokaryotic signaling, and that's why I'm breaking this chapter up into two components. The signaling pathways are quite complex, and I didn't want to cram it all into one lecture. So look for the additional resources in the module, particularly for G protein coupled receptors and examples of those. And I'll try to track down some other examples of receptor tyrosine kinase other than the epidermal growth factor.